Hello friends, welcome to BMH Learning. This video deals with Omni-C kit. The Omni-C kit has optimized DNase 1, a sequence-independent endonuclease to reproducibly digest chromatin, delivering all the characteristics of a high-C approach without the sequence bias inherent to restriction enzyme-based high-C approaches. The kit generates enriched long-range cis reads and more complete contact matrices when viewing chromatin conformation and looping interactions. Omni-C data gives you the most complete genome-wide view for all your genome assembly, phasing, structural variant, and SNP research. The kit lets you dive deeper into the mechanisms of action to help you find answers. The improved coverage of Omni-C libraries enables genome-wide SNP calling, generate high-resolution data that can detect large structural variants, and deliver chromosome phasing information with low switch error rates, all with one library. This was all. Thanks for watching.